We can, as people file in, go ahead and get started. Hi, my name is Catherine Silliman, and I'm a co-organizer of the MarineOmics Working Group, which is part of the Research Coordinated Network on Evolution and Changing Seas. And the goal of our working group is to promote reproducible and robust genomic research in marine science. And to do that, a group of mostly early career researchers have come together to develop recommendations for various omics analyses with the goal of disseminating these recommendations through a dynamic website resource that the community can add to and help grow, since the state of genomics is rapidly changing. And so also as part of this, we're hosting these panel discussions, which are meant to bring in folks to discuss using their expertise these different omics methods. And the discussions are really meant for audience members who are new to these analyses and are just trying to start out and want to start out on the right foot. But also, more expert users who are also trying to figure out better ways to work with these data. So the panel will, I'm sure, get technical at times, but we also, when we get to questions from the audience, strongly encourage introductory level questions for those who are just getting started. We will be keeping an eye on the chat. So if something isn't clear, post a comment in the chat and we'll address it. If questions come up, post it in the chat and we'll either try to work it in or we'll get to it at the end. And then before I turn it over to our moderators, I am just going to throw in the chat a little link to a poll, just a demographic poll, just so we know sort of who's participating if you haven't filled it out yet. And also just our website so you can keep track of what we're doing. And with that, I will turn it over to Sara Wuitchik and Sara Schaal.
Thanks so much, Catherine. Hi, everyone. Thank you so much for joining us today in our discussion about considerations and guidelines in genotyping with whole genome sequencing data. We are very lucky to be joined by some wonderful panelists today who I'd like to introduce before we get started. We have Noah Reid, an assistant research professor in molecular and cell biology at the University of Connecticut. Noah's research involves evolution of pollution in pollution tolerance in fishes, local adaptation in killifish, and comparative functional genomics. We have Tim Sackton, the director of bioinformatics in the informatics group at Harvard University, whose research interests include the evolution of innate immunity, the genomics of convergent evolution, as well as developing best practices in bioinformatics. We also have Nina Therkildsen, an assistant professor in natural resources and environment at Cornell University. Nina's research includes the genomic basis of fisheries-induced evolution and local adaptation, reconstruction of the past with ancient and historical DNA, as well as delineating biologically meaningful management units. We also have Jeffrey Good joining us from the University of Montana, where he is an associate professor in wildlife biology, whose research includes everything from speciation and sex chromosome evolution to population genetics and comparative genomics. And last but certainly not least, we have Misha Matz, a professor in integrative biology at the University of Texas in Austin. His research program addresses questions around adaptation of rebuilding corals under climate change, genome-wide genotyping and gene expression, as well as developing genomic methods in non-model systems. So thank you all for joining us here today. We're very excited to get your input and feedback and expertise on some of the considerations we'll be covering. But just to kind of get everyone, I guess, on the same page, I would really love to hear some sort of things to consider that you think about when you're starting out with whole genome sequencing data, whether, I guess what the definition of, high versus low coverage is in your respective fields, what resources you like to have available before you go into a project, when do you need to make a reference genome versus being able to use a closely related species, those types of questions and considerations.
Should we just jump in? Are you going to call on us?
No, I mean, I guess, yeah. You feel free to answer as many or as few questions as you feel comfortable discussing. And you can either raise hand either on video or with the Zoom reaction if you'd like.
I guess I can go first then. Or Jeff, do you want to go?
No, go ahead, Nina.
Well, I guess in terms of the low versus high coverage, I would consider low coverage anything coverage that is too low to reliably call individual genotypes. And that reliably call is obviously, there's no set threshold. It's going to be a somewhat arbitrary cutoff. But I would think of anything sort of less than 4 to 8x, or I mean, I generally think mostly of less than 4x, something in that neighborhood as low coverage, where I would never try to call genotypes. I would only work with genotype likelihoods, and then higher than that. I would consider medium, and then maybe only high coverage about 20x or something.
Inked people seems to think that less than 10x is low coverage.
I mean, there's not a precise threshold, right? I mean,
Sure.
You sort of transition from, I think, well, there's a paper from Hans Allegren's group looking at PSMC, which is probably one of the absolutely most sensitive methods to genotype error. And they argue, 15x coverage is kind of the minimum you need for good results. But, with 4x coverage, you often don't have very much missing data. So
Yeah,
it's all.
I think a really critical thing to think about with all this is that really with everything, both the approach on what kinds of data you generate, because I saw there's some questions about reduced representation or whole genome or whatever. It all depends on the sort of question and the nature of the question. So if you're pursuing things that are very sensitive to calling SNPs, and especially low frequency SNPs, and you're using an approach that is sort of with low coverage sequencing, it's important to remember that, if you have, say, 4x coverage, that's just usually the median of the distribution. And so some sites will be quite high. And if you just restrict your analysis to those, maybe that'll be useful. But, you sort of think about high coverage in my mind, high coverage is the point at which you get to most of your sites being sort of called with confidence, right? So the Poisson distributed, random coverage generated from whole genome sequencing, most of that distribution being up high. But it really depends on the question. Like phylogenetic questions are pretty robust, low to moderate coverage, figuring out tree shapes and variants in tree shapes. But if you start to think about, say, frequency spectrum based things, or molecular evolution, where you're sort of hard calling genotypes with confidence, and then downstream, that's more sensitive. But it's really context dependent.
Go ahead, Misha.
Yeah, I think maybe it's the next chapter in the discussion, but I just like to say that in my opinion, the biggest difference is not the coverage of the genotyping, but the type of question you are asking in your study. If you're asking the question, which is genome-wide average type of thing, allele frequency spectrum, population structure, demographics, heterozygosity, something which basically needs to have one number for the whole genome. Reduced representation genotyping is the thing. That's what you want to do. And there you can possibly go even high coverage if you want, but that's a question number 2. However, if you want to look for interesting places in the genome, if you want to basically do a genome scanning application, find places in the genome which are doing something different from other places in the genome, whole genome sequencing. There is because if you do reduced representation, somebody will have to come back and redo it. So yeah, but that kind of simple thing for me, at least.
Yeah, maybe before we move on to that, it might be useful to also just kind of give a little bit of things you need to think about before you start this kind of project, right? So no matter what you're doing, and I would argue, even if you're doing reduced representation, you need to think about, you're going to get these little short reads, and you need to align them to something. Now, technically, you can do RADseq without a reference genome, I think the performance is substantially worse than if you do have a reference genome. So
Depends on
you
what
need,
do.
sorry?
That depends what you do. Population structure totally works.
Well, it's not to say it doesn't work, right? But it's always, there's always a benefit to having a reference genome, right? Whether you can get away without having a reference genome depends on your question, right? But again, going back to Jeff's point, you know, you got to think about your question, but you also need to think about, you're going to think about whether you have something to map to, and that's going to influence your experimental design and influence the type of questions you can ask and whether you need to produce a reference genome. Just having a reference genome doesn't always get you that far, depending on what your questions are. If you want to like do any sort of, you know, looking at evolution of protein coding genes, looking at McDonald-Kreitman type tests, looking at, you know, differences between, you know, amino acid or like pi n, pi s, like looking at non-synonymous variation compared to synonymous variation, you need an annotation. You need to know where the protein coding genes in your genome are, which can often be orders of magnitude more difficult to get than a genome itself in terms of work and everything like that. And you need to think about, you know, sampling, right? You have some number of samples and then you have an Illumina run, and how you're going to sort of allocate samples to runs. If samples are really hard
Great, thank you all so much for your input. Those are all great things to consider, and hopefully have everyone kind of on the same page as we go forward in our discussion today. I think kind of building off of the idea of the quality of the reference genome and whether you have a reference genome available or not, with the limitation of having a reference genome in your particular study species or closely related species, especially for low coverage, can you talk about some of the pros and cons of investing in high coverage versus a reduced representation approach?
I guess there's a couple questions there, right? There's the question of what resource you use and when you need your own de novo genome versus using a close genome. And I think a lot of that depends a lot on, from my perspective, we spent actually a lot of time developing tools to utilize a sort of close reference. So we call the pseudo reference approaches. And we have some programs to do this, and we've sort of, you know, it works pretty well. Reference bias is pervasive, no doubt. It affects everything from your estimates of heterozygosity, but really sensitive to more fine-tuned analyses such as gene expression related to allele-specific expression, or if you're, you know, really going into the details of genomics, reference bias is a real issue. So we usually use, even when we're using a closely related species, we usually iteratively map to it and create a pseudo reference that is sort of to the population specific dynamics, but that only gets you so far. And you get to this point where you end up with this sort of trade-off, as Tim alluded to. You know, the hardest part is the annotation, the hard-fought annotation, and even your best, you know, Markov models to estimate gene models are not that good. You have a lot of transcriptome data and a lot of gene models, and it still takes a year of careful annotation. I'm sure Tim has this experience as well, and Nina. And so I think we try and utilize close references, most importantly, to deal with the annotation and what the genome sequence says, or even distant references and sort of integrating across that. And I think that that's a really important consideration as for how you deal with it. There's the reference bias issue, which I think is the thing most people think about, is like, can I use this reference? That's actually not that hard to overcome for some things, although indel variation's tricky always. But like you can map to sort of divergent references and you can sort of iterate your way out of that. But the annotation is a really critical thing to port over. And I think there's always the question about where you start a study, but ultimately we like to fully understand systems. Usually we don't usually always reside at that sort of first skim level of, you know, pop structure and how things are related, but you ultimately want to understand the details of evolution. You got to understand the sort of genome more clearly. So I usually start a project by building all the tools I can, and maybe the first waves would involve reduced representation or whatever, but always with the eye towards developing a system more fully.
We'll go with Misha next, and then Nina.
One of my favorite applications of genomics is doing allele frequency spectrum modeling. And for that, mapping to a sister species reference is actually a very good idea because you get polarization of your SNPs. You know which one is ancestral, which one is derived. So that's a great advantage. And if you're just working with one species mapping to another species, the reference bias is maybe not that much of a problem. It's like you don't give anyone advantage over any other things in the data set. So the question is, I mean, how much lower your mapping efficiencies are if you're mapping to a sister species compared to your own? Tom, what would you say? I mean, there is a threshold, right? So what is a closely related species? How closely it is? To me, it's basically if you don't lose too much, like 5% of total mapping efficiency, it's good enough. No?
I mean, so of course the issue is the mapping efficiency is not uniform across the genome, right? And you may be preferentially losing, you will be preferentially losing reads from the most variable and potentially most interesting parts of the genome. So there's trade-offs, right? But yeah. I'm actually going to plug Jeff's work, right? Like Referee, this program, you know, seems to work really well, and I think there's always advantages to doing that because you may actually not change the, like 80% of the genome may be no different whatsoever, but if you can boost mapping in the 20% most divergent part of the genome, I think it'll always help. But there's no, like, you know, hard lines, I would say.
And the thing that we've noticed is that the mapping efficiency sort of overall parameters, it can be quite subtle differences in sort of number of reads mapped. And a lot of these, especially now, that's one thing that's really improved over the last 5 years is the sort of rapid short read mappers that aren't quite so sensitive to divergence, right? And but you do, the summary parameters do get underestimated. So even with 1% sequence divergence, which isn't even that far of a reference really, you'll sort of systematically underestimate, say, genetic divergence, DXY, or some sort of standard statistic like that, or, you know, and pi as well, if you use a divergent reference without accounting for that. And you can account for it, but we've shown that, you know, your estimates can be off by 20 or 30% with not even that far of divergence. But it's a systematic bias, and as Tim says, it will be non-randomly distributed across the genome, and so often we're interested in rapidly evolving regions of the genome. And so, you know, just sort of being mindful of reference bias and mindful that that for most things, sort of summary statistics overall, and even like overall performance of mapping numbers, don't necessarily cue you into where you're losing and how you're losing sort of subtle biases, right? So I don't know. I'm persistently paranoid about most of these things, and it usually helps, but maybe sometimes it's just paranoia.
So Jeff, up to what level of divergence do you typically have good experience with doing this pseudo reference approach?
Well, I mean, we've gone pretty far, you know, multiple percent. For example, our snowshoe hare work has relied on mapping to rabbit, which is pretty divergent, maybe 12 million years and maybe 5% or so. And we've repeated all that with de novo references, and we get the same results. But the thing that changes, if we just map right to a reference, it doesn't work well. But what we do is we map to the reference, we call genotypes, we change the reference, we map again to the reference, we call genotypes, we change it, and we iterate until it goes static. And that takes a while sometimes. And of course, that doesn't work in the really divergent areas that Tim was talking about. Like you'll have blind spots, but that can work out to sort of, moderate levels of divergence, especially for gene sequences. It actually works pretty well because those tend to be pretty conserved, but.
Go ahead, Nina.
Well, yeah, so the point I wanted to make was also just that if anyone feels that it sounds really intimidating to need a high reference genome, to be aware of how far technology has come in terms of making that a pretty feasible thing to do, to generate a high quality genome. So I think for anyone working with vertebrates, and needing a genome, I would really consider collaborating with the Vertebrate Genomes Project. So we did that for a recent project where basically we have a shared interest. We need a reference genome. They need the gold standard reference genome for their comparative genomics work. And for a very modest investment from our side, we got a full, beautiful chromosome level assembly for a new species. So I would really consider that. And then also there are different commercial services that offer these with Hi-C and PacBio. So at this point, it actually is a pretty modest investment to get a high quality genome. Of course, you don't get what Jeff was talking about, the annotation and all that then requires some manual creation and a lot of work. You don't get that immediately, but at least just for getting the contiguous reference sequence, at least for species with relatively simple genomes, that's actually not so much of a barrier or obstacle for most new projects at this point, at least in my experience.
Thanks. And Noah?
I actually just wanted to ask Jeff a question about that approach. Is there any worry about like enforcing variation that comes from mismapped reads with the way that you do this like iterative updating of the reference? Like you have reads that you're mapping to a non-specific reference genome, and you have reads that go to the wrong spot, and then you just change the genome so that those reads then are attracted to that spot.
Yeah, well, I mean, I guess if you had repetitive areas that were only represented one place, and you weren't, so you use sort of hard and fast genotype, conservative genotyping calls. What we do is we do a mixture where we also remove the areas of the genome for which we have no confidence in genotyping. So we do soft masking, we call it. Well, not our word. That's what it's called. And so we do hard masking where we totally remove it depending on what we want to do. So we sort of do some mixture of confidence and places where we, and so you use things like mapping quality. And if something's mapping many places or multiple places in the genome with sort of uncertainty, then we would just treat that as unknown and remove it as much as possible. But of course, the best thing is to build your own genome, but I feel like it so far hasn't let us. I mean, it's worked really well for lots of things where we've sort of repeated it with de novo genomes, and nothing's ever changed. But the nature of the questions we do tend to be, we're looking at continuous variation. So a couple mismapped reads here and there, it's not a big deal. But so you look at the sort of how things change sliding across the genome. It works pretty well.
Yeah, Tim?
I'll just add one point to what Nina was saying about the assembly, although I don't think we want to get too derailed by assembly, but it's an important point because lots of people are thinking about these projects. VGP is great. I think it's it's modest on the scale of things, but if you want to do a lot of species, it can add up. There's other cheaper approaches, even if you're not working in vertebrates. We've had a lot of success with nanopore assemblies, which, for a few $1,000s, you can get a decent genome, can be very, very cheap, and, applicable to lots of weird things. We do a lot of work on plant genomics that, can be very, very challenging because they're 80, 90, 95% repeats. There's a lot of technology advances out there, and both Nanopore and PacBio are continually improving to the point where there was just from Nanopore, there was just a quote for $10 human genome assemblies, which I don't know if I believe that or not, but if that's the case, right, you're talking like incredibly cheap for a lot of things. And you know, genome assembly is I won't say it's a solved problem, but it's becoming a lot easier than it was if you haven't thought about it for 5 years. Annotation, like what you do with the assembly, of course, is still a challenge.
Okay, I just want to bring in some of the questions that we have from the chat. There's quite a few of them at this point. And so the first question that we got was, do you not recommend any type of RAD-seq or GBS for GWAS studies nowadays? So that's going off of some of our earlier discussions about reduced representation versus whole genome sequencing.
Yeah, I have a strong feeling that no, you should not use reduced representation for GWAS.
Yeah, I'd agree with that. I don't see any reason you would want to do that.
Yeah, I think the scientific question of is something associated with this phenotype is not an interesting one anymore. And that's the only answer you get with reduced representation, not what it is, right? And what it is what we care about. So like it can be a good, we use RAD-seq for genetic crosses mapping panels still. That works really well, both because you have the power of linkage to sort out genotyping error, but also because it's just a really fast way to generate large panels or to fill in additional genotyping on top of a whole genome context. And so that kind of association, I think, but outlier analysis or GWAS, well, we don't use that for that.
Go ahead, Misha.
Yeah, I just want, since Jeff mentioned linkage mapping, I actually would like to underscore this, that RAD is fantastically good for linkage mapping. We just recently, Zach, my collaborator who did our Coral genome, did all sorts of fancy high-C analysis to put the scaffolds back together. We already have a previous scaffolds based on linkage maps. And turns out there's no improvement. It's like exactly like it. So it works. I'm like a now big believer of RAD and good old linkage mapping. So it's one good way to actually put your genome together.
Recombination is always your friend.
Yeah.
If
you're working in a species where you can easily generate large panels like that, right? Like many species, of course, that's quite challenging. The snowshoe hare linkage map is probably not coming out anytime soon.
We've got 3 crosses done, Tim. We've got 3 crosses. We're working on it.
In MarineOmics, though, it's pretty easy because most of our guys generate millions of offspring of each cross, and you can do linkage mapping in F1 if it's outbred population. That's what we've done. So it works.
But definitely the correspondence and linked read sequencing, I think, super powerful for pulling, if you don't have long read where you have contiguous sequences, but you have sort of genomes that are sort of, highly fragmented in small contexts, it is very helpful to pull those together with some sort of physical or genetic information about associations. And I think, the genetic maps are great, but if you can just do a high-C library or some other, it's a shame that 10x Genomics quit doing their linked read libraries, which were quite nice for this as well. But I think that kind of association is really nice. And we found good agreement between those kinds of approaches.
We deviated from the question.
Go ahead, Tim.
I was going to ask, what was the question? But just a general question about like, when do you use reduced representation versus whole genome? I guess I would emphasize, I think, the point that Misha made earlier, that if you're scanning across a genome, you don't want to scan across only part of the genome. So you really, anything where you're doing scanning across the genome really is going to benefit from, doing whole genome sampling. I will say, you got to think about the genome, right? Like if you want to do population genetics of axolotl, I think you're going to have to do RAD-seq for the foreseeable future. You've got a 30 gigabase genome that's full of repeats, like, you know, whole gene, even low coverage whole genome sequencing is going to be very expensive and hard to manage. So there are caveats, right? Like there's certain genomes that are refractory to best practices, I would say, in some sense. But you know, that's not to say RAD-seq never has a use, right? I think, you know, if you're doing phylogeography or something like that, it can be perfectly good. You know, again, I would go back to what I said at the beginning about sampling, right? Like RAD-seq gets more valuable when you have an enormous number of samples and, you know, the cost to sequence them all starts to add up. If you don't have that many samples, I guess I would argue there's never really a reason to do RAD-seq, because if you're getting, you know, you don't, RAD-seq where you have 1000x coverage of each loci is kind of pointless. You may as well have just done whole genome sequencing of those samples.
We've also sort of I mean, I know reduced representation becomes synonymous with RAD-seq, but we've also done a lot of exome capture as well, which behaves beautifully. It's expensive, and it's sort of, we're at this point where we're not developing exome capture for many of our systems now, just because the whole genome approach has sort of begun to pass it by. But one project where we are doing whole exome is we have a project where we wanted to understand, you know, genome evolution across 300 species of rodents spanning, you know, a common ancestor to 20 million years. And, you know, you just can't do whole genome sequencing for that with any confidence. So we've done exome captures across that full span and generated really nice high coverage data of the genic regions, and that's worked really well. So it has those kinds of things have a place. I think that enrichment's always going to have a place because it's a tool that has applications that can be useful. I just think it'll become a little bit more niche, or won't be the sort of, I always thought of RAD-seq as like the gateway tool to genomics, right? And anymore, we're starting most genome projects with low coverage whole genome as a sort of first pass, but there are certainly lots of good applications for those kinds of things. It just won't be the sort of begin and end of the approach, I think.
I'm gonna call in Nina quick and then move on to another question so that we've got quite a few questions to get to today. So Nina, go ahead.
Yeah, I just wanted to say there's kind of this assumption that RAD is always going to be cheaper than whole genome sequencing, but I think it really pays to look at the actual costs that you'll have to pay. Because I mean, we're doing whole low coverage whole genome sequencing now for like $20 to $30 per sample. So depending on whether people are preparing their own libraries or whatnot, it's often now, I think, at a very similar price point. So that should be taken into account too, as people are planning new studies.
Yeah, this is where genome size becomes important, right? You don't have a huge genome, I don't think, right?
Exactly. Yeah, we're like in the nice list, 800 megabits,
yeah, right.
1 GB level, very comfortable.
Yeah.
Yeah, that's of course a huge consideration.
Okay, I'm going to move on to a question that we got from Ben we've touched on aspects of this already, but just to get the thoughts from the rest of the group, he asks, if we want to characterize populations at the genome scale, interesting regions from tens to hundreds of populations, and want to capture the variation that exists in long-term utility for panel development. Should we aim towards higher coverage greater than 20x with few samples, like 10 to 20 samples, or low coverage 7x with many samples? Misha.
Wait, I did something wrong. Wait a second. Well, so yeah, what I already put in the chat is that there is one way to combine the two. If your recombination rates are not ridiculously high, that's where your recombination is your enemy. You can actually make a sort of reference panel out of smaller fraction of your individuals. Say, this is an approach which Zach Fuller, my collaborator, did for our Coral in a recent paper. They sequenced 40 individuals representing the populations at very high depths So basically make sure that you get all the haplotypes done. And then the other few hundred, he sequenced at very low depths and imputed the haplotypes. So this actually works pretty well. Well, if you can convince yourself that you can actually impute with confidence, and it's pretty easy with, you know, validations, cross validations. So this would be great way to get like really whole genome sequencing data for pretty cheap.
And Nina?
Yeah, I think again, it depends on what questions you're interested in, but I'm gonna do a little shameless plug here, because we recently have written kind of a guide to low coverage whole genome sequencing, where we're comparing kind of the trade-offs between number of samples versus the depth per sample for estimating different types of population genomic parameters. And the key conclusion is that for most parameters, you get much better accuracy of inference by doing more individuals at lower coverage than doing fewer individuals at high coverage. And for the particular reason that if you're doing fewer individuals, you're going to have much more sampling error, right? And just getting a few samples. And as long as you appropriately account for uncertainty and work with genotype likelihoods instead of hard calling genotypes and introducing noise, then you actually get, for things like allele frequency estimates, I would definitely go for low coverage. And on this thought on thinking about long-term utility, I'd say thinking 5 or 10 years. Right now we're limited to short read sequencing, but probably 5 or 10 years from now, we'll have link read sequencing, we'll have long molecule sequencing. So I think about what's used. Of course, it's important to be able to use data downstream too, but I think more about what we can learn right now and keep in mind how technology is likely to change in the future. So actually maybe preserving, archiving sample is more important so that we can go back with more powerful technologies in the future. And for archiving sample, having bigger sample sizes, I think would also be an advantage.
Yeah, Tim?
Yeah, I'd also say, you know, from the long-term utility standpoint, I do think there is a lot of value in having a highly confident reference panel for your species of known SNPs. There's a lot you can do if you have that you can't do otherwise. And I mean, you know, the experience of the human population genetics community is a great example of this, right? Like, you know, there's tons of methods out there that that are developed in the context of human population genetics that depend on a reference panel, known SNPs that exist. Like even, you know, basic parts of variant calling pipelines depend on having known SNPs for your species. And so I think that's an important consideration if you're talking about systems where you're trying to develop resources for a research program, that, you know, you can't generate a high confidence reference panel with 4x, 6x coverage data, right? So I would kind of second what Misha said about that strategy of investing in high coverage sequencing for a subset of individuals to develop that reference has a lot of potential. You know, I would also second what Nina said about long read, you know, technology, right? I would say we're a year away from, or 6 months or 0 months away from routine use of long read sequencing for population genetics. I mean, the costs are coming down really fast. The accuracy is going up really fast. You know, there already are some PacBio resequencing data sets available. Nanopore is really pushing their sort of immediate real-time sequencing for you know, human genetics. Most of COVID sequencing, at least in the UK, is done on Nanopore anyway, right? So, you know, that's another consideration. That technology is available now, and, you know, you might think about that as part of a reference panel as well, right? Of course, the accuracy is lower, but you're going to uncover a ton of variation that you wouldn't be able to see otherwise.
Nina, did you?
We often use this. I'm sorry, I just sort of
go ahead.
Agree with everything everybody said. We often use this approach. I was just thinking about this. We have 3 projects we're sort of early stages on right now, and like it appears to be like my go-to move would be like sequence 80 low coverage genomes and 5 moderate coverage genomes and 1 high coverage reference genome. That seems to be the thing that's going on in the lab right now. And, you sort of get broad, you get all the stuff, right? And maybe even sprinkle in some reduced representation, things like exome capture, or even RADseq can give you also those sort of SNPs, as long as they're placed in the context of a genome. You can sort of get panels of SNPs that are informative for things like calibrating your error rates and your variant calling that Tim was referring to. But yeah, sort of a mix of different ways of knowing and different types of estimating parameters, and there's so many different levels of question there.
And I know, Nicholas, you've had your hand up. I think you have a comment on this topic. If you want to unmute yourself and go ahead.
Are we allowed to ask questions like this?
Yeah,
okay.
so.
I just have a very specific question about the Caro paper. I might have interpreted this wrong, but it sounds to me that there were about 50 individuals that were sequenced to high coverage and then are used to form this reference panel, right? And then the authors validated the genotyping accuracy by using the individuals that are already included in the panel. And I found that to be a little bit circular. Is that what happened with that data?
No. You basically, you take these 50, actually it was 40, I believe. You take these 40, you withhold 5, develop the panel from the remaining 35 and try to predict those 5 that you withheld.
Okay, that
so
makes
it's
a lot
like
more
really
sense.
hold out proper.
Thanks.
Yeah. Mm-hmm.
Okay.
Okay, and Ben, you have a comment?
Great, thank you. Thanks for this and for those answers to that question. Just to follow up briefly, yeah, so basically the long-term future use that we're thinking of here is, like there's a lot of funds involved in these types. Like a lot of us are facing this question, hundreds of thousands of dollars going towards whole genome resequencing in the next few years, right? And so, I mean, it's really critical that we understand, where to put those funds towards. And so the long-term use is more like we're trying to build resources for the whole community. And so it's going to be beyond like the immediate questions we're asking of the data. So maybe we want to do like genomic vulnerability or something of the like. And but we also want to have these sort of resources that can go into the future and be used for a decade to come or longer than that. And I know the field changes pretty rapidly. I really like this idea of the tiered approach of, as people have been describing, of a few individuals sequence very deep. Just to follow up on that, so this is my question, if we have like 50 different reporting units, for example, in this species, like unique populations that we would assign to, just for example, would we have to do like deep sequencing on each of those, like do 5 individuals or something at 20x, something like that, and then expand out? Okay, and then low coverage, do another 80 individuals or so, and that would kind of get us a good estimate of that. Yeah, I guess maybe we don't know. But okay, so five deep sequencing, and then what we can do for the other ones. Okay. That's great. I guess not much of a question there, but thank you.
Thanks. And given that our working group is focused on best practices and ways to inspect our data as we go through these pipelines, do you on the panel have any sort of general advice on points between the FASTQ to VCF generation where you focus efforts in testing specific parameters or maximizing genotype estimations? Particularly, are there parameters that you focus on when you're dealing with low coverage versus high coverage data?
Let's name our favorite filters. My favorite filter is min-in, basically genotyping rate. A site needs to be present in at least 50% of my samples visible. Otherwise, it's probably contaminant or something like this.
I would recommend, I mean, one thing that I would recommend is even before you get to the point of calling SNPs, look at plots of coverage, right? Just look at histograms of coverage, look at the distribution of coverage across chromosomes, like, you know, especially if you have, you know, going back to something Nina mentioned at the beginning, right? If you have a kind of spotty genome, right, you'll often see whole scaffolds with no coverage, right? Which should make you stop for a minute and kind of wonder what's going on. Or there's a piece of the mitochondria that got assembled but didn't get annotated as mitochondrial. You'll see some scaffolds with bizarrely high coverage, right? Like it's always just worth taking a minute to look. You know, you can understand things about the sex chromosomes in your system that way. I would say that's a very good spot to stop and just kind of look at the data for a bit.
I think it's also, I just sort of sound like a grumpy old man here for a second, but one of the biggest problems I see with people just starting out with genomics is that they place too much emphasis in the sort of point estimate summary statistics that come out of the mapping rate, whatever rate, you know, like mean coverage and things like this. And usually something always goes wrong or something's never quite right, especially on the developing system. And it's amazing how much, like, people will spend so much time trying to filter with filters and look at the distribution of the sort of output summary statistics. And it's amazing how much you can learn by just actually opening up a genome browser and looking and seeing what your data, I know you can't do that across the whole genome, but like just actually look at your data, look at how it's mapping, look at the variance, look at when you're calling SNPs, whether or not you believe them, and a little bit of that eye training, even just a week or so of sort of exploring the data just makes a world of difference because, you know, everything about picking filters, so much of it is so context specific and arbitrary in many ways. And you're trying to convince yourself about the truth of the biology underlying it. And so I think always just taking a moment to really understand what is going on and not be too beholden on just pipelines is a pretty important step of just getting in any system, you know, regardless of the approach, low coverage or high.
Great, thank you. I will do Nina and then Noah.
I think Noah was best.
Okay. I just wanted to I agree with everything Jeff said. I kind of want to second Tim's statement about coverage, especially when you're doing a lot of low coverage sequencing. A lot of the sort of outputs of variant callers become way less useful, you know, like freebase and GATK output in the info field, like 50 different statistics you could potentially filter on. But if you have like 1 or 2 reads per sample per site, then like none of that's going to help you much. You're not going to get significance for whether or not the variant is always at the 3' end of a read or always on the, you know, forward or reverse strand. And so like I wind up paying a lot of attention to coverage, whether things are too high or too low. And I want to say one other thing, something that I do, which is kind of, I think of it as kind of hacky, but the ancient human genetics people do it a lot, is with low coverage data is pseudo haploidize the low coverage data, just pick one allele per locus. And that means you're running your data through like a standard variant caller. But if you're using something like one of these standard variant callers, you really want to make sure that there isn't some kind of Hardy-Weinberg prior in place on the allele frequencies, because that can really just like basically make up genotypes for you. And so if you can turn that off, you really want to turn that off. So that's just as far as parameters that I worry about.
I think another thing to be really aware of that I only discovered very recently is how sensitive low coverage sequencing can be to batch effects. So if you're, for example, now that we are archiving data and sequencing platforms are changing so quickly, you might end up having part of your data set being generated on one Illumina platform and the next being on a different. So we had that issue with one of our recent projects. So we ended up having data from, some of it was from a next-seq and some of it was from a high-seq. And we just and they also had different read lengths because we had different types of data. And we had just naively at first analyzed it just as a single batch. But then when we started looking into it, we realized that there were these huge effects of having these different sequence data. So we had to go back and implement all kinds of diagnostics to discover how just the different mapping, how we could lose alleles. We didn't pick them up in certain batches because we ended up filtering out reads that had mismatching alleles in certain regions, and also with some of the Illumina platforms adding these poly G tails when basically no signal is the same as a G. And even some of the tools that are meant to remove those didn't fully remove those tails, and also gave us a lot of false. And this was so this is especially with low coverage data, I think is particularly sensitive to that. But that was definitely really eye-opening for me to see how big of an impact that could have.
Go ahead, Misha. Oh, Misha, you're muted, so.
Really like to second Jeff's point that eyeballing the data is always a great idea. It's like whatever you do, eyeball the data any possible way you can. Also on pseudo-haploid calling, it's a really cool approach, especially for low coverage data. And Angst has this IBS matrix, which is essentially pseudo-haploid calling. So it's very useful for if you want to do population structure. It's my favorite way of determining genetic distances. It's very robust to coverage variation. Also for filters, there is one interesting thing which we kind of came up with at some point, but it did not catch up in the community. We did, since we use RAD, we can afford replicating at least some of the individuals. We did replicated genotyping of like 5 individuals in the whole data set. And these you can use to actually fine-tune GATK filters. You know, GATK has this wonderful base quality, no, not base quality, I forgot what it is. It's machine learning algorithm which determines
Variant quality score.
Variant quality score recalibration. You can train them based on these replicate genotypes which you know are true. So that's the way for you to get the true SNPs, because those are the SNPs which are truly repeated, like heterozygotes which are repeated between replicate genotypes. Those are definitely true, and you want to recover those. We find that's very cool. I mean, at least the best allele frequency spectra I got were based on these things. Well, so one idea.
Noah, did you have another comment?
Yeah, Nina's comment reminded me that I have had a lot of like bad experiences with batch effects arising from differences in coverage and read lengths and stuff with low coverage sequencing. And in my case, maybe this is something that won't apply to a lot of people, but I found that, so the fish that I work on have very high genetic diversity. I guess the Minidia does as well. But there's a lot of variation in the amount of genetic diversity among populations. And those kinds of batch effects are non-random with respect to how much diversity is in a population. So if a really high diversity population with really high diversity has far more rare variants in it. And so when you lose those rare variants, you're losing more of the branch length off of that population. And so these biases can be really insidious in that respect. At least I think that's what's happening. I don't have like very high coverage data as a gold standard or a panel to prove that that's what's going on, but it seems very likely. So these kinds of biases can sort of creep in all kinds of weird ways that you might not predict a priori.
Sure, thanks, Noah. Tim?
Just wanted to say one more thing while we're talking about parameters to be aware of. I think it's also important for everybody to just be aware that GATK is written from the perspective of a human geneticist sequencing 30x coverage human genomes in a large factory context. And if you go into it for your 1x coverage, low coverage data set or something like that, you will often get very poor results unless you change things. So there's a bunch of sort of hidden parameters in GATK that have to do with how, what data it will throw out, like how many reads are needed in order to even bother looking at a particular segment of the genome. And the defaults of those are tuned around this 30x human genome. So if you don't change them, you'll throw out a ton of your genome without you even really realizing it. If you use GATK, if you go with the approach of like, running through GATK to produce a VCF that has genotype likelihoods and then loading that VCF into something like angst, as opposed to going from BAMS via angst, which has its own set of complicated issues that are difficult to resolve. So that's just, want to throw that out as something everybody should be aware of.
Yeah, that is a great point, always to think about how these pipelines are developed and what their test data is on. Along those lines, in developing panels for species of interest, what would you recommend for low versus high coverage whole genome seq in regards to confidence in your genotype calls? Is this dependent on the genomic resources that are available for the species?
Sarah, can you please repeat the question? I'm not sure I understood it, to be honest.
Yeah, of course. If you are developing a SNP or say a genotyping by thousands panel for your species of interest, is there recommendations for how confident you are in your genotype calls if you're using low or high coverage data? And is that dependent on the resources that are available already for your species of interest?
So it's the question about how you select those markers or how you genotype them?
I mean, you could answer both, but genotyping would be the first thought.
Well, I think again, it then comes down to what your goal is. If you just need something that has to do with allele frequency, then doing more individuals at each at lower coverage should give you eliminate some of the sampling effects of doing fewer individuals. So that'll give you more accurate estimates. If you're really interested in inference that is heavily dependent on accurate identification of rare alleles, then you probably want to go a bit higher, but at least our simulations suggest that after about 4 or 5x, you should be able to reasonably accurately, at least be able to estimate to GMSD and Watson theta and things like that. And then having larger sample sizes is then advantageous for some of the other types of inference you want to do.
And kind of along the lines of, I guess, considering these different types of data with the programs and ways that we analyze them, how well do low coverage programs deal with things like demography, inference, and identifying local adaptation, considering these types of programs are relatively new? Yeah, Misha.
This is my best pastime to apply low coverage sequencing, to do demographic reconstructions based on low coverage sequencing. But to be honest with you, I'm super dubious. There is really no good validation for these reconstructions. So the only validation that I'm aware of, and I'm only aware of because it's my own paper, so when we did the demographic reconstruction of migration rates in the Great Barrier Reef between 5 Great Barrier Reef populations, and they aligned almost exactly with what the currents would do. Otherwise, I mean, how else can you validate population size changes back in time or migration rates? So this is a big question, and I love doing this. And there's the math and the program seems really awesome, and the pictures are great. But do we trust this? I would like to hear everybody's opinion on that, actually.
Go ahead, Tim.
So I'm just gonna, throw out a place that, maybe doesn't get enough attention, but probably should get more attention is, I mean, the human ancient DNA community has sequenced more low coverage samples than like probably most of the rest of us combined, right? There's a huge amount of data that is being generated there. And that's pretty much all low coverage. I mean, that's changing, right? Now people are sequencing, some of these samples with like capture approaches and things to higher coverage, but a lot of that is very low coverage data. And I have to admit, I haven't kept up with that literature as well as I probably should, because it's so vast, and it all gets published in Nature and Science, and you get a one-page advertisement for 200-page supplemental methods, and it's kind of a lot of work to actually figure out how they do their analyses. But I think for anybody who's kind of starting out, that's a valuable place to look in terms of finding approaches. And of course, in terms of validation, right? Obviously, validation of evolutionary, validating evolutionary history is really difficult in general, right? But in terms of populations whose history has been studied with non-genetic methods, ancient humans is the absolute best you're going to get, right? So, there's definitely, some, I think, lessons to be learned there, which unfortunately I can't report on because I haven't read the hundreds of pages a month that are needed, but just want to throw that out there.
I would just add on top of that, just some interest in the ancient DNA of humans world. I think the most surprising thing, the thing to keep in mind when we sort of talk with confidence about reconstructions of evolutionary history at whatever scale, but particularly if you're talking about population history and growth and contraction and things like that, is there's a couple major issues that people are coming to terms with. I think the ancient human data have shown us that you can do a snapshot, reconstruct current sort of population history going back tens of thousands of years and come up with a robust model. If you sample individuals from the landscape from 40,000 years ago, many of them are not genetically represented in the current samples. The complexity and turnover of just, even something as simple as the human population migration history, which is actually not that complicated from versus lots of things, just tells you that how much is absolutely missed with a retrospective reconstruction. It could be you don't care about that because it's no longer represented, but it's a little bit, don't be overconfident even with robust estimates of that you're sort of actually reconstructing the full evolutionary history of these lineages, because it's incredibly complex and drift is really extensive in a lot of these circumstances. I think the other issue that people are really coming to terms with, and especially you see this with a lot of, if you look at some of the the sort of single genome estimators of demography, the various programs that will sort of estimate your growth curves and so forth. They all look the same, right? And they all, like, you don't even need to estimate it. You can just draw it by hand ahead of time. And just because it's largely tracking other things such as link selection and the effects of purifying selection and how that affects genetic variation. And I think that we're still coming to terms with how link selection influences estimates of demography. And I don't think it's hopeless to estimate demography. I think it's a critical part of any kind of outlier inference, but take it with a grain of salt and realize that there are many other processes that are resulting in allele frequency changes and structure across the genome that have to do with selection and aspects of that.
Tim, and then Misha.
I just want to highlight the point that Jeff made, which I think is so important, about the fact that it fundamentally arises from coalescent theory, right? Like what we're sampling now is not representative of what's in the past, right? We're sampling the descendants of the one individual by chance who survived, the pieces of the genome, right? And we know from basic principles of population genetics that is not going to reflect the population that was in the landscape 50,000 years ago, right? That's like one individual that was in the landscape. And yet, like, we try and reconstruct all these histories and things, and we're basically just assuming that the population, that this individual, that the descendants of this individual are representative, and human ancient DNA shows that's like fundamentally untrue, right? There's just waves of population replacements, which must be happening everywhere, right? And I think it really sort of calls into the question, like, how much we can learn about what was happening, what was actually on the landscape 100,000 years ago from current day genetics.
Go ahead, Misha.
So, yeah, this is definitely true that we're not reconstructing populations as history based on the place where we sampled them. We're reconstructing the history of that lineage that we sampled. That's true. Actually, yes, all these demographic reconstructions, especially population size back in time, might be not reconstruction of population living in this particular village back in time because it could have been replaced, but at least it most likely was replaced. But at least it pertains to the lineage which you are looking at. Whatever it was, whatever it was doing, that's its genomic history. Also, it should not be taken at face value in terms of population size changes. It could be, for example, periods of introgression from another species, which look like a great increase in genetic diversity, which not because population size changed, but because the influx of things from another species. And things like that, like linked selection could look like a bottleneck. And that I would also like to add that originally these demographic models were developed based on neutral expectation exclusively. And now they put in parameters which are explicitly supposed to account for selection scenarios, such as background selection and differential introgression across the genome, which is supposed to account for location-specific selection pressures. Super interesting field, and even less we know how reliable these things are. But yeah, super interesting to play with. This is a bicycle, which is very interesting to try to learn to ride.
Great, because we're running out of time. I'm just going to ask maybe one more question from the chat. That seems fairly quick, and then we'll wrap up. So Katie Lauderhouse asked, can you identify deleterious mutations from low coverage whole genome sequencing data? Do you have thoughts on that?
Katie, are you asking about specific mutations, or are you asking about the distribution of fitness effects? Well, I guess I would just say that the specific mutations are always sort of hard with low coverage data, because if they're deleterious, they're usually by definition at low frequency as well. But I think you can do a pretty good job with low coverage data if you're careful at estimating the distribution of sort of differences in the site frequency spectrum of different functional classes of data, and make inference into questions about, for example, how much genetic load there is in a population, and in issues like that. I think that there's things you can do that are estimating a full distribution, but identifying specific deleterious mutations might be difficult, given that they tend to be low frequency. But the site frequency spectras will shift, if you're looking at non-synonymous locations versus, other regions of the genome, for example.
Great, thank you so much. We are going to be wrapping up in, I think our deadline is about 5 minutes, and this discussion today has been really incredible. So thank you so much for everyone who's both attended and is on the panel today. We really appreciate your time and your input and expertise on these questions. I'll do a quick reminder to everyone that this recording is going to be available through the website for the MarineOmics Working Group, which is marineomics.github.io. And we also have 2 panels upcoming, one on June 4th on population genomic analyses, and one on June 18th with RNA-seq data considerations. Both of those you can use through the same access link that you registered for today. And as we wrap up, I'll pass everything over to Catherine for some closing comments.
Nope, you got all of them. Yeah, thank you all so much for coming. Someone asked, will the videos be on YouTube? And they are on YouTube. I'll throw in the link to the RADseq one. And yeah, they'll be up, probably give us about a week to get that all wrangled, and we'll put them on the website. But thank you all so much. And that was a great discussion. Really appreciate you all coming. All right. We will see hopefully everyone in about 2 weeks.
Thanks, everyone.
Yeah, thank you.
Thanks, that was a lot of fun.
Yeah, great
Catherine,
job,
there was some-
Sarah. Oh, please.
Oh, sorry. I was just going to say there was some really great questions and discussion in the chat. So I'm going to export that as well, just in case there's some follow up, or if people want to follow up with some of those questions later. It was so active, and there were so many awesome people on this call that it was, well, impossible to get through all of the questions, but just in case we want it for some follow up.
Yeah, no, yeah, I was just about to do that, but I think it would be better in your guys' hands, because yeah, no, I think that there's a lot of good, just survey of what concerns are in this chat.
Mm-hmm.
But yeah, no, great job, guys. That was awesome and exciting and a little fun and heated at times. So I think it was exactly what we were hoping to get out of this. So you guys did a really great job.
I mean,
I also
yeah,
found it became surprisingly profound in some